Hi, this is Rethik, your biology subject matter expert from MemoNeet. And in today's video, we will be concentrating on concept of dominance, like what exactly the concept of dominance is. So in the previous videos, we have discussed two characters, right? One is dominant and recessive, and how that been expressed from one generation to the other generation. So in this concept, we will be concentrating on what exactly this dominance is and how it is going to express itself from the other traits or characters. See, coming to the concept of dominance, the concept of dominance in genetic refers to the relationship between the alleles of a gene and how they influence an organism's phenotype in sense. So how they are representing the characteristics and how they are influencing one specific organisms on their phenotype or their entire physical appearance and even internal appearance, everything, their observable traits, their characteristics or their traits are nothing but their physical appearance or biological appearance. Everything is being how they are being you know, expressed and how they are being influenced. So that is what we are going to see. Alleles. So what do you mean by alleles then? Alleles, because here I have told you like alleles, right? Genes can have different forms that are called as alleles. For example, a gene for flower color might have red allele, so will be represented as capital R, and white allele will be represented as small r. So in alleles, there are again two types of R. One is that is capital R, and another one is small r. So the first one is the dominant allele. So what do you mean by dominant allele? An allele is considered dominant if its effect are fully expressed in the phenotype. That means if the effect or if it is dominant, it is expressing its effect completely, fully in the phenotype, even when only one copy is present. Okay. For instance, in a genotype of R and R is nothing but only one copy is present, capital R and small r. So here only one copy is present, but still in F1 generation, what you are saying, only red flower is what you are saying. That means now this allele, is expressing its character completely. For instance, if the genotype of R and R, one is actually the red and the another one is white, but still the flower would be white because the red allele here is dominant. The next we have is recessive allele. Say like an allele is recessive, it's is if its effects are masked. Masked is nothing but screened. Okay, by the presence of this dominant allele. Say like capital R and small r, where small r is not expressing its character. It has not completely vanished because in F2 generation we are going to see its character once again reappearing, but in F1 generation, it is been masked or it has been what? So this have been either, not expressed or it is masked in one generation because of the presence of what? This dominant allele. So in our example, we have taken that the white flower, color, which is represented by the small r allele, would only appear in the phenotype if the organisms have two copies. That is, if the organism has only R and R, then only it will be appeared. So if not, it won't appear. If there is only individual phenotype like small r and capital R, then it won't appear, right? Only if it has two copies of recessive allele, then only it is going to appear. So the next one we have is homozygous and heterozygous. So now what is homozygous and heterozygous? An organism is homozygous for a trait or character if it has two identical allele. Say like if it is allele is capital R or small r. So both the alphabets is same. We normally call them as homologous or homozygous. Sorry. Getting. So what is homozygous allele here? So homozygous is nothing but an organism in homozygous for a trait if it has two identical alleles. One is capital R, or small r and small r. If it has two different traits, alleles are expressed too differently, like capital R and small r. So we normally represent it as, or we say it as heterozygous. Heterozygous. Okay, so this is difference between homozygous and heterozygous allele. So there is another dominance called complete and incomplete dominance. What is complete dominance here? The dominant allele completely mask the effect of the recessive allele. So we normally call it as complete dominance. Say like in red color or capital R and small r. So your capital R is completely masking the character of small r. And what as a result, the capital R character has been expressed. That is, resultant will be the red phenotype. Okay, so then we can call it as the complete dominant. Incomplete dominance, well, like what happens? So here, neither the allele is completely dominant, leading to a blended phenotype. Say, like if it is capital R and small r, instead of expressing red color, so another new blend character has been expressed, say like pink flower. Okay, say like what? Pink flower. So here one thing that I can see here is instead of this is the dominant allele and this is the recessive allele. So according to me, the dominant allele should express its character and mask the recessive one. But here what is happening? A mixed or a blend color has been expressed. So let's say like pink color. So the parent had either the red color flower or white color flower, but the offspring are blend colored flower, that is the pink flower. So we can call them as incomplete dominance. So we'll see about like how incomplete dominance. Okay. So in this genetic phenotype, when two true breeding parents with the different traits are crossed, their offspring display a phenotype that is blend of both parent traits rather than one complete masking the other. That means capital R is a dominant one. According to me, it should mask the small r character. But here you are been seeing the expression of the blend character. For example, we have what capital R here, which is RR, and white color flower plant, which is small r and small r. So resultant offspring will obviously be what? Capital R, and offspring might be small r. So resultant will be what? Completely different. That is the blend character is what you are seeing. That is the pink flower you are seeing. So this is demonstrating that neither the allele is fully dominant. Okay. So neither the allele is fully dominant. Where you are seeing this in the F1 generation. So when this F1 generation is self, okay, then you will see the ratio of 1 is to 3 is to 1. Sorry, 1 is to, yeah, 2 is to 1 ratio. So where 1 will be what? Red color, and another two will be the pink color flower, and another one will be the white color flower. So this intermediate expression illustrates how alleles can interact to produce a new phenotype, which can often lead to the diverse range of traits in the population. So this is actually an example of the snapdragon flower. Okay, so this is what incomplete dominance actually happens. You have this. So what happened is there is completely red color and white color. So when crossing is happened, okay, as a result, what you're seeing in F1 generation, blend character is expressed. Say like pink character is expressed. Selfing matlab capital R gamete has been self-crossed with the same. So as a result, what you're going to get now? So you're going to get 1 is to, so this is like 2 is to 1. That is, 1 will be the red color two will be the pink color, and another one will be what? White color. Okay. This is what you're going to get in incomplete dominance. Again, let us take another example in incomplete dominance. Okay. Say like considerably. Let's take an example of gene that contains an information for producing an enzyme. So there are two copies of the genes. Okay, that is nothing but two allelic form. Let us assume as like a normal allele produce a normal enzymes that is needed for the transformation of S substrate. Okay. Therefore, the modified allele could be responsible for production of one thing will be if the allele is modified, what all it is going to produce? The normal or less affected enzyme and a non-functional enzyme or no enzymes are produced. Okay. So, in the first case, that is the normal or less effective enzymes produced, the modified allele is equivalent to the unmodified allele. That means less effective enzymes or normal enzymes are still produced. The enzymes which actually helps in the transformation of the substrate, yes. So it is still producing. So it will produce the same phenotype trait. That is, the result in the transformation of substrate S will definitely happen. Such equivalent allele pairs are very common. But if the allele produced what? A non-functional enzyme, okay? So, or no enzyme. Non-functional enzyme means the enzymes which will not be responsible for the transformation of substrate, or if it doesn't produce any enzymes at all. Okay. The phenotype may be affected. The phenotype trait will only be dependent on the functioning of the unmodified allele here. So if the allele is unmodified and allele is coming, then only the function we are going to see. The unmodified or functional allele, right, which represent the original phenotype in the dominant allele, and the modified allele is generally what it is showing the recessive allele. Okay. But if non-functional allele or no enzymes allele are produced, we can say it as incomplete dominant because of some modification in the allele. So we are finding what? So non-functional or no enzymes formation allele. So this is all about the incomplete dominance part. I hope it is very clear to you all, right? So we will move to the next topics, that is inheritance of two gene. So what do you mean by inheritance of two gene here? The Mendelian also worked with the crossing pea plant that differs in two characters. So one was yellow color and round seed. Okay. So one seed was what? Yellow color. The seed was yellow color and it was what? The round seed. And next was green color wrinkled seed. So another one thing was green color. So it was something like wrinkle shaped. Okay, like this. Wrinkle shaped. So these are the two pea plant seeds he have taken. So Mendelein found that the seed resulting from the crossing of the parent had yellow colored and round shaped seed. Thus, yellow color was what? Dominant here, and the green over the green color. And round shape is dominant over the wrinkle one. So this result were in identical to those that he got when he made a separate monohybrid cross between the yellow and a green seeded plant and between the round and wrinkle seeded plant. As a result, you can see here, students, that round yellow is there and wrinkle green is there. In F1 generation, he got round yellow was what he got. That means round color and yellow round shape, and yellow color is dominant over the green color, wrinkle shaped. So when in next generation, when selfing was made, he found that when Mendeleev made a self hybrid in F1 generation, as you have seen. So in F2 generation, he saw again some difference, like round yellow were 9 and round green were 3, wrinkle green were 3, wrinkle yellow were 3 and wrinkle green was 1. Say like the phenotypic ratio. Hmm. Bartini Ajji. Phenotypic ratio was 9 is to 3 is to 3 is to 1. So this is what at the end product I have got.